Welcome, everybody. Welcome to today's Timothy Johnson Medical Scholars Seminar. Before the introductions of today's speaker, I'd like to remind everybody that this particular seminar program, sponsored by the Fralin Biomedical Research Institute and the Virginia Tech Fralin School of Medicine, is named in honor of Dr. Timothy Johnson. Dr. Johnson was one of the founding faculty members of the VTC School of Medicine. He was instrumental in working on developing the curriculum on this research-intensive medical school education paradigm, and was very much committed to this idea of scholarship and having visiting physician scientists be part of the program and provide these opportunities to the medical students and the graduate students. So it's a good time to remember Dr. Johnson and all his contributions. Today's speaker will be introduced in a moment by one of our own investigators, Dr. Zhisheng. Dr. Sheng will introduce Dr. Yung from MD Anderson Cancer Center. And Dr. Sheng, I think, as most of you know, is himself a cancer researcher, and in fact, a brain cancer researcher here at the Fralin Biomedical Research Institute, and somebody who knows the field well and is very knowledgeable of our guest, Dr. Yung. So without further ado, I'm going to turn it over to Dr. Sheng for the introduction. Jake?
Thank you, Mike. It's my great pleasure to introduce today's speaker, Dr. Alfred Yung, a world-leading neuro-oncologist. Dr. Yung attended medical school at the University of Chicago, and he conducted his internship and residency training at UCSD, and his chief residency at Cornell, and his fellowship at Stoke Catering. And after that, he joined the Neuro-Oncology department at the MD Anderson Cancer Center, where he has worked for 40 years. He has served as Deputy Chair of Neuro-Oncology and Medical Director of Neuro and Supportive Care Center in 2000. In 2001, he was co-director of the Brain Tumor Center, and he's a Department Chair of Neuro-Oncology at MD Anderson for more than 16 years. And he's currently the Senior Advisor of Brain Tumor Center at MD Anderson. And Dr. Yung and the team have been conducting both laboratory and clinical research to find treatments for glioblastoma, the most deadly form of brain cancer. For example, he led the study for FDA approval of Tomozolamite for treating glioblastoma and the registration study for FDA approval of bevacizumab for recurrent glioblastoma. His research also helps understand the biology of this disease. For instance, the cellular signaling in glioblastoma stem cells. Throughout his career, Dr. Yung has been published more than 377 peer-reviewed articles, and he served as editor-in-chief of Neuro-Oncology from 2007 to 2014, and he still serves as editorial board member of Neuro-Oncology. Dr. Yung won numerous awards. To name a few, he has been named as a top 1 doctor in America since 2012, and he was awarded with the Lifetime Achievement Award by Society of Neuro-Oncology in 2015. Dr. Yung's research has been continuously funded by the NCI, together with his fellowship with National Foundation for Cancer Research. He has been co-chair of the NCI Brain Malignancy Steering Committee since 2011, and is a special advisor to the CEO of the National Brain Tumor Society. And Dr. Yung was also named to the Blue Ribbon panel of experts advising the National Cancer Moonshot initiative. So we are so excited to have Dr. Yung to visit us today, even though it's virtual. So without further ado, please join me in welcoming Dr. Yung.
Thank you, Dr. Sheng, for this very nice introduction. My mother probably will smile in heaven, and my father will not believe what you said. But it is an honor. Now, before I go further, let me check, make sure do you see my screen?
Yes, we do, but you need to go into presenter mode.
Okay, now I'm going to presenter mode. And
do the swapper.
Okay.
That's perfect. Thank you.
It worked. Okay.
Yes.
So I said it is an honor for me to join the seminar program, even though it's virtual. And I know that we tried to do it in the last year so that I can come in person. I always enjoy, visiting place in person. As I said to Leslie earlier, but hopefully COVID will be under control, hopefully by the end of the year, and then we can resume traveling and resume some face-to-face meeting. But on the other hand, I think COVID had changed the way that we conduct research and we conduct conferences, and we also would change the way that we meet, from face-to-face to more virtual. But I think it's all good. It's all good. I also really enjoy meeting the medical student, just now for lunch, and we have a very nice conversation. Now, for this seminar, what I thought I would do is, first of all, I like to make it kind of, informal, since this is a program for the medical student, and I understand that you have a small elite group of students, and I think Leslie also told me that there's a strong research emphasis in the curriculum and allowing time for the student to research. I think that's really exciting to know. Being a physician scientist myself, I'm always a champion of research and discovery, so that we can move the envelope further to the edge, and keep it further, and also educate and train the next generations of physicians and scientists, that we can continue to make progress in the disease that we're interested in, not just cancer, but other disease, like being a neurologist and neuroscientist, degenerative disease of the brain, multiple sclerosis, all these top issue that we have. So here's my disclosure. Just very short. I chose, I'm for my career in medical school, I chose to devote my research time and thinking in brain tumor, partly because of my fellowship training, but partly also because actually my mother died of a ependymoma, and that kind of provides some impetus, some for me to go into brain tumor modeling, CNS tumor modeling, and treatment research. I pretty much devote my research career on glioblastoma in the last 30-something years. Glioblastoma is the most common, but also the most malignant tumor, the primary tumor of the brain. And it is also, unfortunately, one of the most invasive and heterogeneous tumor, or solid tumor, compared to all other solid tumor. The median survival is pretty bad. We can even use the word dismal. Compared to other cancers. Patients with a glioblastoma have a median survival or still hovering around 14 to 18 months. And I put this slide up, again, like, Dr. Sheng already told me that I've been in Anderson for 40 years. When I started, after I finished my training, and I started really as an attending, the graph on the left is a graph to show you at that in the 1980 New England Journal of Medicine, as a composite analysis showing that the median survival of patients with glioblastoma with the treatment of radiation therapy and BCNU chemotherapy is 11 months. And 25 years later, 25 years later, another article published in the New England Journal of Medicine in 2005, and with the report on the upfront use of temozolomide combined with radiation therapy, the median survival is 14.6 months. So you can see that in 25 years, we moved the median survival of this tumor for patient from 11 months to a 14.6 months, a 3.5 months increase in 25 years. Now, even though I must say, the increase in median survival is a little disappointing, but on the other hand, we have seen quite a bit of improvement in the quality of life for patients because of the improvement in surgery, improvement in radiation therapy, and even improvement in chemotherapy care. Wait a second, how come I cannot move? Yeah, sorry. Okay. Why we cannot move the needle further? Now, this is a list. This is a list of challenges or a list of reasons that we face, in terms of why we could not make things better for our patient. If you can see this whole list of reason, it's the top of the list is basically tumor biology. The heterogeneity of the tumor, and also basically the genetic makeup of the disease, and also the change over time with this genetic makeup and heterogeneity. And then the second most top problem is because of the biology of the tumor is very resistant to treatment. And the resistant come with the genetics, and other the resistant change with the treatment and with time. And this becomes besides the biology of the tumor, the next big problem that we face with treatment for tumor in the brain is the blood-brain barrier. God created the blood-brain barrier to protect the brain from toxic material. Toxic material circulating in the blood cannot penetrate the microvessels into the brain parenchyma, but that also become a obstacle for drug delivery to tumor in the brain, and that is a major problem. Also a major problem because most of the drugs that are being developed to treat cancer are treating systemic cancer, and systemic cancer has no blood-brain barrier, so they don't want the drug to get into the brain to cause brain toxicity. They want to keep the drug in the systemic circuit, in the systemic organ to treat the cancer. So the drug that's available, for cancer use, even for brain tumor use, do not cross the blood-brain barrier, and that is the issue. Now, talking about heterogeneity of the tumor, this slide shows you the Wxo classification. In the old day, classification is based on morphology. How many cells you see, how ugly the cell, how many vessels you see, how bad looking the vessels are, and whether there's any necrosis or not because of the vascular circulation of the disease. These are become the feature that the pathologists use to classify the tumor from less malignant to more malignant. So from low-grade tumor astrocytoma, to mid-grade tumor as anaplastic astrocytoma, to high-grade tumor glioblastoma. But since 2013, when NCI sponsored a large scale effort to look at the genetic background of the tumor, the Cancer Genome Atlas (Tcga) project, and the first, good for us in the neuro-oncology field, the first cancer that was chosen as a pilot study is glioblastoma. And so in 2013, we see the report showing the molecular landscape or the molecular background of the morphologic heterogeneity that we see in morphology. And this is a list of the genetic mutation that you see in the whole study actually includes 500 tumor. And this is the partial list, the first publication to show you the number of genes that's highly frequently mutated, and there's also a large number of genes that is less frequently mutated, rarely mutated. But on the end, that also tell you the noise level of the genetic background of glioblastoma. It is really noisy. There's a lot of gene involved. This is further. This effort was further analyzed, and now we are this is the second publication from the Tcga analysis of 500 glioblastoma, and look at all tumor level, low grade to high grade, and we come up with this. Now we call the molecular classification or molecular subgroup to supplement morphologic diagnosis. And in a way, nowadays, when you have a surgical removal of the tumor or biopsy of the tumor, the pathology report will not only report the morphologic diagnosis of whether this is a astrocytoma, oligodendroglioma, but it also will report the molecular feature behind it, whether there is involved 1p19q mutation or involve 1p19q deletion, whether we see specific gene mutation like p10, PIK3CA, or amplification of EGFR. These become the key feature to tell us whether this is a astrocytic tumor, oligodendroglioma, whether this is a glioblastoma with feature that is going to be very malignant, or that feature with a different gene mutation, whether it is more responsive to therapy. And so it become a must that we have a molecular background diagnosis. This is even makes things more complicated. The next publication that came out from Europe, when they studied thousands of tumor together with the methylation platform. Now, not only look at the mutation or the fusion protein, but in this effort, they look at the methylation profile, epigenetic pattern of the tumor. It actually they come out with 91 classes of the tumor. So now you see, the difficulty that we face in terms of creating treatment that is effective. So now, I don't have. I think it will take us another lecture to talk about the influence of heterogeneity in tumor biology and resistance, and where we go from here. I thought that I would address the second most important issue in terms of lack of progress is the resistance of the tumor. And when we talk about resistance to the tumor, I just listed here. Nowadays, with the advance in molecular biology, and also not only genetic profiling of the tumor, and now we're moving into single cell profiling. Now we can look at the tumor environment cell by cell. Now we begin to understand that tumor resistant to tumor is not just the tumor cell itself. And it's also the environment is another major factor. Even when the tumor, we can see that there is several ways that the tumor become resistant. One is that the intrinsic, the molecular background, the molecular landscape of the tumor cell. And then as we treat the tumor, the tumor genetic or tumor molecular background evolve, change, adapted to the insult from the treatment, and either continue and kind of mutate to overcome the treatment effect. So you have the adaptive and acquired resistance that come with treatment. And then also, not only when we provide treatment to one single target, there's always another target that the tumor can use, or another pathway the tumor use to escape the attack from the drug or from the treatment modality that you use. And so that the tumor cell can escape the treatment from utilizing parallel signaling. And then the tumor environment comes in, the immune cell become a major factor. How the immune environment modulate the tumor resistance, and how the immune cell change, secrete or provide, activation or suppression of the killer cell that can kill the tumor and provide surveillance. The stromal cell that used to provide nutrient support to the tumor, it could become a major factor on the nutrient need and the metabolic need of the tumor, and creating an environment of resistance. And with newer single cell data, we also begin to know that in the brain, now the tumor can actually interact with the neuron or neuronal system, and the neuronal system become a shield, or the glial system become a shield to the tumor. And now you can see how complicated that is to deal with the environment. So the goal of my lab, is focus on what as I shared with the medical student earlier, in research, we can be focused, or we can also attack multiple areas at the same time. So there are several areas of research that I have in the lab. One of the areas that we have is really focusing on the resistant mechanism of the tumor. We focus more on the tumor environment as opposed to the immune environment or the stromal environment. And one of the focus that we spend a lot of time on is how to make the standard therapy work better. The standard therapy meaning radiation therapy and chemotherapy with temozolomide. Before temozolomide came out, we used a lot of BCNU. BCNU and temozolomide actually belong to the same category of drug. They are alkylated. They work on the DNA, and let me see. They work on the DNA and causing damage to the DNA. In terms of temozolomide is a methylator. It adds a methyl group to the guanine, and also the adenine, and but in different location. Now this slide shows you that the three areas that temozolomide add in the methyl group. The major path is adding methyl group to the O6 guanine. 70% of the action of temozolomide is making the OSIC methyl guanine. And the other 30% of the time, it add the methyl group to the N7 and N3 adenine. And then you go through the different level of repair mechanism and the cell use to repair the damage. But if the damage is not repaired because of lack of repair enzyme or lack of the homogeneous recombination or mismatch repair gene, then the cell, the single strand break become double strand break, and then the cell die. And this is how it works. So we try to look at different way of saying, well, how can we make temozolomide better? Now, I just want to also comment on the kind of system that we use. As we always think about research, finding, a drug or finding drug target. It very much depends on the model system that you use to study research. Now, in our group, we spend a lot of time in the early year using established cell line. In the neuro-oncology community studying GPM, the most common established cell line used is U87, U251, LN229. These are established cell line, from one tumor, and they've been passed along for tens of years. And so, but that's been the system that's been used for a long time. The system that we use nowadays, more in terms of more recently, is we now was able to culture more stem cell-like tumor cell. They're called stem cell tumor cell, and these cells that we extract from the tumor, you can extract them from individual patient's tumor, and they actually maintain the genetic feature of the patient tumor closer than a cell line like U87, U251 that actually been passed along for a long time. They lost some of the critical molecular feature of the human tumor. So we chose to use the glioma stem cell model so that we now look at cell that is closer to the genetic makeup of the tumor. And these so-called glioma stem cell can be, they come in sphere form. And so you could look at the pathology of the sphere, and you can also look at the growth potential of the sphere. And you can also put the sphere into the mouse brain and generate an in vivo model. And that the pathology of the in vivo model is also closer, similar to the human tumor. We generated in our group, one of our investigators had spent a lot of time generating a large number of glio or GSC, we call it glioblastoma stem cell clone. And then we also spend a lot of time sequencing them and create the molecular makeup profile of each of these lines, so that we know exactly what is the genetic makeup of the line and how well they compare to the tumor that the line comes from. And so now we think that this is a better system because we have a bank of line and with a well-characterized molecular background. And as we use them to look at drug effect, radiation effect, and look at the change before and after treatment, we can always, go back to the molecular background to find out why it works and why it does not work. So I chose to look at 3 projects. And so let me go to the first project. for the first project, the goal of the first project is to find out what is the parallel signal that the tumor cell utilize to escape the activity of temozolomide. And one way we chose to look at this is that we use the interferon RNA system, to try to knock out a gene, and see whether by knocking out that gene is removing that signal will synergize the toxicity of temozolomide, and whether that signal is the escape signal that the cell use. And you can use a whole bank of siRNA and look and knock out a large number of genes at the same time, and then screen it compared with the one that treatment versus no treatment, and look at the RNA that disappear. Because if the RNA is the siRNA is stopping one gene, and if the gene is the critical gene that temozolomide uses as escape, then there will be more cell kill, and that siRNA will disappear from the treatment group. Now, this is the cartoon that tells you how it works. If you look at this is the siRNA library that we use. They have multiple siRNA, and when you put it into the cell, incorporate the cell, it will get incorporated into the cell, and knock out, presumably will intervene with the transcription and the expression of that gene product. And then we divided cell into two groups: one, no treatment with temozolomide; one group you treat with temozolomide. Now, if the siRNA knock out certain gene, and that gene is required for survival of the cell after temozolomide treatment, then it will go away. Then the temozolomide will kill the cell, and that SI will disappear from this pool. So basically, what you're looking at is you look at the pool here and subtract the pool here. The siRNA that survive here, that means the gene that survive here, is the gene that helped temozolomide. Now, this is using siRNA. I think you probably know that with the coming of CRISPR knockout technique, knocking out the DNA instead of knocking out the RNA, you have a much more complete removal or knockout of the gene with CRISPR approach. So this technique of synthetic screening with siRNA, it pretty much nowadays is replaced with CRISPR, the library, using CRISPR screen. This library that in our experiment of using temozolomide as a drug and look for synthetic target that help the toxicity of temozolomide. When we do the subtraction of the pool, we come and rank the gene that disappeared the most, or the gene that is enriched in the control side, but not in the temozolomide side. And we see that there are 5 genes that had pretty much enriched in the control well, but not in the treatment well. And the next step you need to do is to validate whether this is the gene. Now, one of the genes that we chose to look at, there are 5 of them, we chose to look at BRCA first. Even though we have screened all 5 of them, but I'm going to show you something that we see with the BRCA gene. Mainly because BRCA gene mutation and BRCA gene loss is a very frequent event in breast cancer and ovarian cancer, and it's also been known to be a candidate of enhancing damaging DNA damage repair by radiation and by cytotoxic drug. Now, since there is no chemical to remove no chemical drug available to really remove BRCA1. We validate the observation with doing, again, using SI approach to knock down the gene expression of BRCA1 and then treat the cell with temozolomide. Here we see that, to show you that, actually we see a variable response. Some cells respond better. Some line, they show you the 2 lines here, GSE11 has a good response, and seeing a combined effect of knocking down BRCA1 and temozolomide. But this is another line, the 23, it doesn't have much effect in terms of the combination of knocking down. So we're not seeing genetic effect here. And this is the way the bar graph to show you the change in the apoptotic cell. Now we can see that if the combination work, there is a synergistic activity of temozolomide and knocking down BRCA. We see an increase in the number of apoptotic cells in the culture, as opposed to the one that we don't see the apoptotic cell. And this is another way to show that the synergistic effect of removing BRCA1 or downregulated BRCA1 with temozolomide is causing apoptosis of the cell, but not causing cell cycle arrest of the cell. Turn out though, the difference between the 2 lines, GSE11 and GSE23, is actually on, again, in the genetic background of the line. If we bring in the issue with p53 gene. Now, this is another gene that involves in DNA repair. Now, the line that we can see the genetic effect, the synthetic effect, now that is GSE11, had a wild type p53 background. Wild GSE23, that when we do not see the synthetic lethality, has had a mutant p53 background. And here just show you that you can see increase with the combined treatment with the siRNA and the TMZ, you see increased DNA damage, as signaled by gamma H2AX. And in the GSE23, you don't see it. And so it suggests to us that the action, the synthetic action, it depends on the DNA, depend on the p53 background of the cell, whether the cell is able to, again, utilize another repair mechanism or not. And this will show you, again, for the student in terms of we need to kind of validate this is the right one. So we take GSE11 and say, well, we're gonna knock out p53. Now, we knock out p53. This time we use this CRISPR approach. Use CRISPR to knock out p53, and so you said there is no p53 expression in it with the knockout. And here to show you that with the knockout, when you treat the knockout with temozolomide, if there's an effective knockout, unfortunately, again, I'm sorry, we use SI approach here and not a CRISPR approach. We see knockdown of gene. So we actually see, when you combine it and you knock down the p53, which is required for the synthetic effect, you see less. You see less combined effect there. And we also see less apoptotic cell. So as a conclusion for this project, which is published 2 years ago, last year, is that with the synthetic lethality screen with the SI library, we pick out 5 genes, and BRCA1 is one of the 5 genes that is able to synergize with temozolomide. In addition to the gene itself, we also know that wild type p53 is required to see the synergistic effect. If the cell has a mutant p53 background, we do not see that. No. So and we see the apoptosis, which is again would go along with that if you are able to really prevent the repair of DNA damage by the Tomozolamite, then you see the cell die in the apoptotic fashion, and not just shut down the cell cycle. This project 2, which is also published in Cancer Research, it shows you that we come in terms of, well, how can we make Tomozolamite work better from a different angle? Rather than looking at the parallel signal or the gene that is required, we are looking now the known repair pathway of repairing the DNA damage by Tomozolamite. How can we shut down the repair pathway? If we shut down the repair pathway, can we enhance the toxicity? And one of the known way, there are two ways that the cell use to repair damage from Tomozolamite, depending on where the damage is. In the O6 guanine situation, there is an enzyme that the cell make called methylguanine methyltransferase or alkyltransferase. This MGMT is the enzyme that come in to remove the O6 guanine damage and allow the cell to be repaired and survive. In the other side, like I said, the 30% side of the damage done by Tomozolamite in the N7, N3 guanine and adenine side is repaired by base excision repair, which again utilize the POP pathway, and the POP pathway help repair the damage, because POP comes in to the single strand damage by the O6, by the methylation, and bring in the repair enzyme through pyrylation of the protein and bring the repair enzyme to repair the single strand break, to prevent the single strand break and to become double strand break. So now, as you may know that in other cancer, which that PARP inhibitor has been used very successfully to enhance the toxicity of treatment, is breast cancer and lung cancer. So we decided to say, well, what can we do with PARP inhibitor? Can we enhance the toxicity of Tomozolamite? Now, I'm going to show you this the first project that is, show you that there is another observation that we made by using, even though our intention of going in to say, well, can PARP inhibitor enhance the toxicity of the cell to Tomozolamite, or PARP inhibitor work in breast cancer by, like in breast cancer, can kill the cell by itself. So we use our GSC library to screen them, to screen the number of PARP inhibitor that is being used in the clinic in breast cancer and ovarian cancer. Again, what we see is that there is a kind of preferential response to the PARP inhibitor among this number of lines. You basically can divide it into two groups. One group is quite sensitive to it with a low IC50, and the other group is very resistant to it with a very high IC50. As you can see, a higher IC50 and low IC50 group. And what turned out that if we stacking again with the advantage of knowing the genetic background of the line that we use, if we stack it up by with the genetic background of the line, line them up this way, then we see actually the sensitive group almost all the EGFR amplified line belong to this group, the sensitive group, as opposed to the other genetic feature like P10 mutation, you kind of scatter around, and P53 scatter around, and so then it kind of give us a suggestion and say, hey, let's look at it. Is EGF receptor amplification, which is seen in about 60% of GBM, could be a marker to tell us that the cell is more sensitive to PARP inhibitor. Now, the next thing we did actually, well, we have a number of PARP inhibitor, and we look at it, they all function as an enzyme activity to pyrylation and inhibit the PARP function. But there is a difference among the 4 that we use when it comes to PARP DNA trapping, because another way that PARP inhibitor work is that it actually traps the complex of PARP and DNA on the DNA. And this complex becomes toxic to the cell besides shutting down the repair. 2 of the 4 that we use have much higher ability of trapping the PARP DNA complex. That is, and also turn out that these 2, talazoparib and pamiparib, the 2 that had trapping at higher toxicity to causing DNA damage and causing single cell activity. And here to show you that, again, in the amplified cell, we can see the DNA damage with the trapping one, talazoparib, but less with the non-trapping one, veliparib. And again, we went into just show you that, again, in research, we need to validate and verify if we think that EGFR amplification is important. What happens if you remove EGFR? Now, can you see the reverse, of the activity? And here we take the line that has amplification of EGFR, GSC 262. We knock out. This time we use CRISPR to knock out EGFR, and so there is no EGFR gene in the cell. And treat it with talazoparib, and now you can see that the cell become much more resistant to treatment. This is the control cell, and this is this is the treated cell, and you see that you reverse the sensitivity, and this this is just show you, can we do a opposite way? Instead of knocking out EGFR, can you make this resistant line with low level EGFR to a high level, express high level EGFR? Can you make the cell sensitive to the PARP inhibitor? Here we take another line, 272, that no amplification, but we amplify it by knocking in the EGFR gene, the wild-type EGFR gene. And you can see that the knock-in with the EGFR wild-type versus the kinase dead, the one that does not have the kinase activity, you can see that the improvement, the resistance that come with the sensitivity that come with of amplifying the gene is only with the wild type. So that, again, to show you that the EGFR receptor signal has to be a working signal before we can see the improvement in toxicity. Turn out the improvement and the mechanism of action is related to reactive oxygen species. Which here we show that in the amplified line, there's a higher level of ROS. And if you treat the cell with a ROS inhibitor, shut down the ROS activity, then the activity of the PARP inhibitor also go down. And the next thing we said, well, if we see it in the cell in vitro, can we see it, the same function in vivo, in animal? Now, so this will show you that when we use a subcutaneous model we can see growth inhibition of the tumor with talazoparib, with the PARP inhibitor, and actually demonstrated also it increased DNA damage in the tumor specimen. But on the other hand, I also say this also suggests, tell us another issue that we have. Even though we see talazoparib work in the subQ model, when we switch to a in the amplified line, in the amplified line, and we do not see it in the non-amplified line, but we cannot see the same activity when we put the tumor cell in the brain, in the intracranial model. Now, we think that the difference between the subQ model and the IC model is that we require the drug to pass the blood-brain barrier to get to the tumor. Talazoparib does not cross the blood-brain barrier, has a very low level, and that's why it worked in the subQ model, but does not work in the IC model. Here's the conclusion from this project. So when we start out to look at the effect of PARP inhibitor, and what we see with a single agent activity before we add the temozolomide is that we actually see some PARP inhibitor work by itself. Those that had high DNA trapping ability work as a single cell. And also this ability of killing the cell also is very selective. It's selective in the cell that has high EGFR activity or EGFR level, the amplified cell. So EGFR could be used as a predictive marker for the tumor that has high level of ROS, high level of DNA damage activity, and also had high reactivity to PARP inhibitor that have the DNA trapping capability. Now, what about combining the PARP inhibitor with temozolomide? Actually, we learned another thing. This is the project 3 that also resulted in a paper in Neuro-Oncology, in the journal Neuro-Oncology. And when we look at the two together, besides that besides C, that there is increased toxicity of combining temozolomide with the PARP inhibitor to the tumor cell, but we see another selectivity of the combination. Now, and this selectivity actually based on the repair enzyme MGMT, whether the cell has MGMT or do not have MGMT. Again, just to show you the role of MGMT in the repair of temozolomide toxicity. MGMT come in to remove the O6-benzylguanine and allow the cell to survive when it removed. If there is no MGMT, then the cell will die because the temozolomide, the O6-benzylguanine, would change the single strand damage to a double strand damage. When there is a mismatch repair gene deficiency, which most glioblastoma has. There is another way that there is one way to remove turnout in looking at the biology of glioblastoma. There's one way that the tumor actually remove MGMT effect. That is by methylating the gene, the promoter methylation method. When the tumor cell has a promoter methylation in the MGMT gene, then MGMT will not express. So when we in glioblastoma, there's another marker that we have to identify up front is whether the tumor has MGMT methylation of the promoter or unmethylation, and it's being split about 60-40. 60% of the tumor had unmethylation, and only 40% have methylation and turning of gene. That means a majority of the tumor is resistant to temozolomide toxicity. When we screen the number of cell, the cell line bank that we have, again, we see two groups. One group is sensitive and one group is not sensitive. And it now turns out this bit it's split into whether the presence or absence of MGMT, whether the MGMT gene is methylated or not methylated. And now, make a long story short, it turned out is that now we discover as we look at using immune precipitation and kind of identify the MGMT from the cell line and what happened with the treatment of PARP inhibitor. It turned out that for MGMT, the methylguanine methyltranslate to work to remove the O6-benzylguanine. If the first level is that it requires pyrylation by PARP before it can bind to the DNA or bind to the target O6-benzylguanine, as demonstrated in this immunoprecipitation gel, and also the graph here will show you pyrylation is required for MGMT. Now, only pyrylated MGMT can bind to the DNA onto the target. If you treat the cell with PARP inhibitor, it removed the pyrylation ability. So the MGMT is no longer pyrylated. And because it's no longer pyrylated, it cannot attach to the DNA. When it cannot attach to the DNA, it cannot remove the O6-benzylguanine. And it's just to show you that with the PARP inhibitor inhibiting the pyrylation MGMT, you see much higher level of DNA damage, or the accumulation of O6-benzylguanine in the cell. With the accumulation of the O6-benzylguanine, you increase the toxicity of the drug. And again, when we go to the animal model using the sub-Q, now we can see the suppression of growth. Now, we do not see this suppression of growth. We do not see this suppression of growth in a methylated line. The reason that we think that we cannot see it in the methylated line is TMZ is very effective by itself when there is no MGMT around. So even though when there is no MGMT around, you cannot improve the activity of temozolomide by removing the function of MGMT. Only in this side, when you have MGMT around, then you inhibit the activity of MGMT, you can see the improvement in action. Now, we can, in this case, we actually can see improvement in survival in the intracranial model. When we use another PARP inhibitor that cross the blood-brain barrier, well, this is olaparib that cross the blood-brain barrier much better than talazoparib. And then we can see the improvement in survival curve when we combine the two together. So in this study, while we enter the hypothesis that we go in to this experiment is to say, combining PARP inhibitor with temozolomide, removing the repair mechanism of the basal excision repair can improve the toxicity of temozolomide. But then we also make a new observation that that PARP inhibitor actually is required for MGMT function, and its PARP inhibitor can be used as a inhibitor of MGMT function, and to remove the repair activity of MGMT, so that temozolomide activity is increased, just like in cell that has no MGMT around in the methylated tumor. So this suggests to us that actually this can be translated into the clinic as focusing the combination on MGMT unmethylated patient, those patients that have MGMT in the tumor, as opposed to the patient that have methylation of the MGMT, that have no MGMT there. So it kind of, in a summary, to show you the journey that we have gone through in the lab about studying, the way to enhance temozolomide toxicity to improve the efficacy of standard therapy. So we go in 3 different ways. One, we look for parallel target, and we find several of them. BRCA1 is one of the several. And when we try to look at, can we inhibit the repair mechanism of temozolomide toxicity? Can we inhibit it? And we identify, a biomarker with EGFR amplification as a biomarker to predict increased activity of PARP inhibitor. And we also identify a new way of inhibiting MGMT activity as opposed to methylation of the gene promoter. You can now use PARP inhibitor to actually remove the function of MGMT by preventing its binding to DNA. So when you go in, again, it's to show that when you go in with a hypothesis, you can prove that your hypothesis is right. When you go in with a hypothesis, sometimes you actually have a collateral or corollary benefit of actually turn out another observation that is very beneficial. But on the other hand, I must say that the task that in front of us is very fast. And as I also mentioned to the medical student earlier, the advancement in medicine research, in medical science, and the advancement in the kind of research that we can do with molecular biology, molecular genetic, immunology is so wide open. So it is actually create a lot of opportunity for us to tackle these, question that we have, these problem that we have in inhibiting progress for us to improve the survival or the quality of our patient. So there's a lot of work in front of us, and I want to encourage the student, that the key is to observe, to look, and to think, and to learn as much as possible, and then decide what you want to do with your life. This is the group of people that work in my lab, and all the work is being done by them. I'm just a speaker, and this is a group of talented people in the lab, postdoc, including Dimpi Ko, Xiao Pei, Xiao Fang, Xiao Long, Zhe. Several of them have now moved to another lab to have independent scientist position. Okay, thank you.
Thank you, Dr. Yang, for his great talk. So I have a couple of questions here posted. So for let's get. This is a question from Shahrukh, the first-year TBM student, and he asks 3 questions. I will come up with one by one, very complicated questions. So the question number 1, what could be the specific role of this new base excision repair independent mechanism in healthy cells, and also in other types of aggressive cancer, such as lung cancer, colon cancer, and pancreatic cancer?
Well, I think taking advantage of DNA damage repair mechanism, is a wide open, but also very complicated, area, because there is so many enzymes involved and a lot of interdependency of different pathway. So, basic, base excision repair mechanism is not a single mechanism because it's also linked with, action of p53. It also linked with non-homogeneous recombination, whether it's governed by BRCA, BRCA gene or BRCA1 like state. So I think this is actually is in a very it is an area that researchers on DNA damage repair mechanism focus on a lot. How do you look at the role of p53, the role of base excision repair, the role of non-homogeneous recombination, and together. So I don't think you can look at base excision repair as a single, way. You really look at the network. Now, if you go to look at the network map, it's quite complicated.
His second question is, how do you compare the effects of temozolomide and PARP inhibitor on glioblastoma stem cells and the non-stem cell tumor cells, non-stem tumor cells?
Well, it has been, I think, if you take temozolomide first, temozolomide actually is not a very active agent in vitro. It requires very high concentration to kill the cell. If you study temozolomide alone, and in proliferating cell, it's a little lower, even though it's high. If you compare proliferating tumor cells, so-called, the other name to call it is probably end-state tumor cell, versus stem-like tumor cell. We always say that stem-like tumor cell is more resistant to drug, including temozolomide, even higher concentration when you look at the stem-like tumor cell versus the proliferating tumor cell. And it's even worse with PARP inhibitor. I don't think PARP inhibitor is active as a cytotoxic drug for glioblastoma themselves. Because a PARP inhibitor require a deficiencies in homologous recombination. So you actually would have to see the PARP activity in BRCA1 deficient cell, which is not very common in glioblastoma. Okay. Now we happen to see that in our work with single agent activity of PARP, but that's only it's only caused by the DNA trapping, the complex, not by modifying the repair, the enzyme activity. So I don't think you will get much mileage from single agent PARP inhibitor.
His third question is, how do you see the effects of PARP inhibitor on normal cells, and specifically the off-target effect on PARP mediated DNA repair mechanism?
I think you know that I do not have a lot of information, because I think you can learn from, since PARP inhibitor is used widely, is approved by FDA for breast cancer and ovarian cancer treatment in patients with BRCA deficiency. And also it's now being studied with combination, especially with PI3 kinase inhibitor. Because it shows you that it does require, inhibition of parallel signal. So I would say, since I do not know the toxicity, not studying the toxicity in normal cell very well, I would say it probably very much the toxicity would depend on the the genetic background, and especially is whether you have a BRCA1 or 2 deficiency background, whether you have genomic deficiency of BRCA, you may see more toxicity in normal cell.
Okay, so we have some questions. I think just got 1 or 2 questions from Samantha Houghton, and she said, "Thanks for a fantastic talk. Have you considered targeting MGMT pyrrolation specifically, rather than using a general PARP inhibitor to avoid issues with PARP inhibitor toxicity?"
That's a very good suggestion. I mean, I think that means biology. And yes, I think then the question is there since pyrrolation is a function of PARP, right? So can you prevent pyrrolation without stopping the activity of PARP? Or the other way to say is that, well, is there other way of stopping the pyrrolation function of PARP protein other than the current group of PARP inhibitor? Right?
That's right. Her second question is, in your first project, what mechanism do you believe is causing sensitivity to Tomozolamite only in p53 wild-type background? For example, does p53 specifically respond to O6-methylguanine mutations to initiate apoptosis?
That I do not know. I think we actually been looking. Our lab is not a big lab in p53 biology, which is again by itself is very complicated. P53, but we have made these observations about the importance of the genetic background of p53 wild type protein activity versus mutant protein activity, which I think we've been seeing is interaction with when we study the P10 PI3 kinase pathway. P53 also comes in. We study the activity of EGF receptor. We actually also see interaction between EGF receptor signal with p53 signal. So I would say that it all probably to do with cell cycle and also the DNA repair activity from an oxidative stress and DNA damage. The requirement of the wild type function. The wild type protein is probably more important than mutant protein.
Great. Now we have about 5 minutes to have the last question. It's also from Samantha, and she asked, "Have you looked into talazoparib drug delivery mechanism that would be effective in reaching gliomas, or would you prefer to use another PARP inhibitor that can more easily pass the blood-brain barrier?"
Well, that actually also is a very good question, because that opened a big area of research. How do if you think that there is a target and there is a agent that work well against the target, but yet it does not cross a blood-brain barrier to get a high enough level into the tumor and having the on-target effect. How do you change that? do you do chemical screening to identify molecule that cross the blood-brain barrier, meaning the molecule may be more hydrophobic, so it cross the blood-brain barrier better, or you can use some chemical modification, put in more hydroxy moiety and make it cross, and that's one area you can do that. And there are also people doing, can I attach it in the nanoparticle and using a special lipid nanoparticle to bring the drug across the blood-brain barrier. And there's also effort to look at with some peptide, amino acid peptide that can be transported through the blood-brain barrier by active transport. So there are different ways of doing that. So it again, I think there's a lot to do is which approach you want to do depending on how good that drug that you have on your hand for on-target effect, if you can bring it across the blood-brain barrier. Yeah.
Great. So let's thank Dr. Yang again for his wonderful talk. Thank you all.
Thank you very much, Dr. Young. That was spectacular and really holds out a lot of hope and promise for getting this figured out for GBM. Just amazing progress your team has made. Thank you. Fascinating.
Thank you. Enjoy it a lot. Hopefully can provide some stimulation to the medical student to enter neuro-oncology.
I hope so. I think some of
we need a lot more talent.
Yeah, that's for sure. I see some of the questions there, Tim, were from some of our graduate students as well from the TBMH program I saw. So I guess, I don't know, when he had lunch, was it just with medical students, Leslie, or a combination?
I don't know.
Medical
I don't
and
know.
graduate students,
Yeah.
I think,
It was a combination.
Right, yeah.
Yeah.
It was good. They were good questions. It was a great seminar.
Mm.
Anyway, thank you again, Dr.
Thank
Young.
Okay,
Very much.
great. So
Thanks, Z.
Yeah, so I think your next set of, I don't have your itinerary right here, but I know you have to switch to a different Zoom link for the next set of meetings.
I have to go back to the,
Mhm.
where's Cameron? So I go back to the first link.
Cameron, is that right?
Yes, that's correct. We'll just jump back over to the first link. So
Go back to the first link. Okay.
Okay.
So we will leave this and then go back.